So like I mentioned before, the Diels-Alder reactions are not entropically favored. So we do need to have heat to make them work. So we need to have low to moderate temperatures. So oftentimes I do these at like 80 to 100 degrees, 150 degrees. But it turns out if you go too high, on the other hand, we will favor the retro Diels-Alder. So very high temperatures, usually somewhere over 200, 200, 250, 300 degrees, you'll get the backwards reaction. So you'll actually break apart your molecule and you're essentially going to get back your product. So just be aware that temperature does play a role in these reactions. If you get too high, you can break the molecule back up. Now, you can also do these reactions, these Diels-Alder reactions, with alkynes. We can take an alkyne and do a Diels-Alder reaction with it. But because I only take one of those pi bonds to do this, the second pi bond is still there. And so we end up with that second pi bond still on your molecule, and so we end up with a cyclohexadiene molecule. And so if you have a triple bond as your dienophile, you're going to have two double bonds still in the structure. All right, so let's look at, when we look at these mechanisms, we don't explicitly see an electrophile and a nucleophile. So what we're going to do is use molecular orbitals to kind of prove which one is going to do our reaction. So I kind of know what the end result is, but I'm going to use what's called symmetry allowed concepts to kind of help us. So if I were to take the HOMO of the diene and take the LUMO of the dienophile, so if we look at the HOMO, right, and we look at the LUMO of each of those two things, because that's all that matters when we're talking about reactions. It's the HOMO and the LUMO that are going to participate. I'm going to focus on the HOMO of the diene, and I'm going to draw this. So remember, two are in phase, and two are not in phase. And if I take the LUMO of my diene, which is this one, notice that when they're not in phase, because we have a node, we can get, and I'm going to, I think I drew this backwards. We can see that the phases are identical. Okay, so there's a good overlap. And remember, to have orbitals overlap, we need to have them in the same phase. And so it turns out that we can have kind of side by side these orbitals overlapping. I'm going to show you what that looks like and what that really means for our molecule. So if we take the HOMO of the diene and the LUMO of the dienophile, we get what's called symmetry allowed, which the orbitals can overlap. So one has to be underneath the other one for this process to happen, and we can form our signal bonds. So these two phases overlapping is what we call conservation of orbital symmetry, meaning we're going to conserve how the orbitals can overlap, and that's called symmetry allowed. And this works when the HOMO of the diene reacts with the LUMO of the dienophile. And if we go back to what we know, HOMO is associated with nucleophilicity and LUMO is associated with electrophilicity. So I actually can figure out what kinds of molecules might work and how they might interact with each other and how I can make them work better by looking at that. And so this goes back to almost everything that we've been talking about so far. And so one thing I do want to point out is that people who do this reaction with butadiene and our ethene, the reaction gives you this product, but it actually gives it to you in very low yield. It's not a great reaction, but what we do know is, since the diene is our nucleophile, if I add a substituent onto my HOMO that makes that HOMO more reactive, higher in energy, this reaction goes faster. And I know that if I put a group on my electrophile, my dienophile, that makes the LUMO drop in energy, I can actually have a better reaction happening. So if my HOMO of my diene, let's say it starts here, and I put something on the molecule that causes the HOMO to be raised, so I'm going to call this diene with a substituent, so with X, or and my LUMO, let's say I start out with my LUMO of my dienophile here, no electrons, and I add some compound, some group onto it that drops the LUMO, then I make this gap easier and make it so much easier for the electrons to go in it. And so you're going to do some classwork related to that in class. But basically, I can accommodate these Diels-Alder reactions by playing around with what substituents are on the molecule, because the HOMO of the diene reacts with the LUMO of the dienophile. So now we know the diene is a nucleophile and the electrophile is the dienophile. So the diene is the nucleophile, the dienophile is the electrophile. And one way that I used to remember this when I was in undergrad, the more pi bonds, maybe more nucleophilic. That's not why, but that's a trick that I used to use to remember that. So we take our electrons and go into our LUMO. In class, you'll do some comparison to what groups you can have there and then determine, okay, if my diene is this energy and my dienophile is that energy, what can give you a better reaction? And so we can favor the types of reactions that we can have. In the Diels-Alder reaction, we can actually play around with what substituents are on the molecule, electron withdrawing or electron donating. But one other place that we see that these reactions are favorable in terms of the groups that are on there is when we're talking about bicyclic compounds. So that's what we're going to wrap up with. So when we have bicyclic compounds, when they overlap with each other, so if I start with cyclopentadiene and I react it with, let's say, I react it with these aldehydes, which is what's going to produce these products down here, you're going to end up with a bicycle. So you're going to end up with these two molecules. And it turns out by having these electron withdrawing groups in our molecule, and when these molecules react with each other, this is your diene and this is your dienophile, by pointing those electron withdrawing groups underneath all the other pi bonds of the diene, it helps to temporarily accept electron density and kind of stabilize your transition state. So and I'll show you a picture of that. What I mean is, as the molecules are approaching each other, if you point the electron withdrawing groups underneath the diene, it helps to temporarily take some of that electron density from its molecular orbitals and temporarily kind of hold on to them. So it's kind of like a crutch. It's kind of like, it's just a temporary relief while this reaction is going on. It helps to stabilize. So it turns out our major products end up being where those electron withdrawing groups are underneath the bicyclic compound. So they're going to be underneath where you see our big bridge coming off. We have a minor product, the endo product, which you can play around with the conditions to try and get more of it, but oftentimes it's actually kind of tricky unless you use some catalyst that can help facilitate this. But what does this look like? So, like I was showing you, if you look at these arrows, as the diene comes in and reacts with the dienophile, those electron withdrawing groups that you have, and remember carbonyls are electron withdrawing groups, they temporarily accept some electron density from that pi network that's above it, and that helps to stabilize the interaction. So it forms a stable pi network temporarily during the transition state, and that it's in that transition state that helps to form these products. So endo tends to be the major product. It helps to stabilize. So if we're talking about stability, then we're talking a little bit more about thermodynamics. Now, there is some kinetic aspect to it. By stabilizing the molecule and anchoring it like that, it can react a little faster, but it's going to be a little bit more stable of a product. Okay, so our endo tends to be preferred because of the stability of that intermediate transition state interactions that happen with those. So if I pretend that my fingers here are those electron withdrawing groups going underneath my diene, them being underneath, helping to stabilize this is good, but because they're underneath, they're going to be pushed down. And as the molecule reacts, you're going to have your bridge and they're going to go down, and that's why they're opposite to each other. And we call that endo. Endo means to go inside, and those electron withdrawing groups are inside the pi network. Exo means they're, let me see if I can point my camera the right way. Exo means that they're outside, and so they're free and out there. They're not helping the reaction out. All right, so here's another image of what that kind of looks like. With the endo is where you're going to have your pi network, your carbonyls being underneath, helping to accept electron density. But remember I said that there is some aspects of this thermodynamic in terms of stability, but also by anchoring, it's going to be kinetically favored. So we're actually having a little bit of both things happening. The only problem with the endo is that in that process, there are some sterics that are associated with that, some spherical issues, but it still tends to be pretty reactive. So it may seem that way because it seems like sterics can play a role, but it's actually as electron withdrawing groups, they help to stay. All right, our very last thing that we'll talk about really quickly is we can have other pericyclic reactions. So an example is a 2 + 2 where we have 2 pi electrons and 2 pi electrons reacting. And we can draw electrons moving around, and we can get these cyclobutanes, which are not really stable. We don't see these happening very much. And one of the problems with this reaction is they are not actually symmetry allowed reactions. So they don't really, you can't just put ethene and ethene together. They're not going to do anything. And so it turns out, if you think about it, if I take the HOMO of one molecule and I take the LUMO of the other molecule and I try to react with them with each other, they are not symmetry allowed, right? However, we can play a little trick. Remember in Gen Chem, when you add light to something, you can cause electrons to move up in energy. I can actually move an electron from the HOMO into the LUMO, and now the LUMO is no longer the LUMO, it's now a HOMO. And because it has that shape now to it, now we have two symmetry allowed molecules. So you might ask yourself, well, isn't the LUMO that's going to accept that HOMO going to jump in energy? And so how we play, it's kind of that same trick where based on the addition. So we take one flask where we add light, and we take one flask where we don't add light, and then we mix them together. And then we can have a reaction where one HOMO of one set and one LUMO of the other set are going to be reactive. Now, this has to happen very fast because as soon as you remove the light, the electrons are going to go back down. All right, so we'll practice a little bit on conjugation and color in class, but one thing I do want to mention is that, remember, your chapter question talks about conjugation and color. The more conjugated something is, you might see different colors. There's more to the story, and we'll talk about that as a problem in class. But if you break the conjugation, like when we do electrophilic addition reactions in Organic 1, we can actually change the color. And this is how many bleaches work for dyes that you have, like in clothes. So if you take bleach, which is an oxidant, remember we can oxidize double bonds to diols. And if we broke the double bond and made it into a diol, for instance, we break conjugation. And that's why we sometimes see color changes when we bleach our clothes or bleach our hair and things like that, is that now we have double bonds that we've broken, and we've broken conjugation. We can actually do this with other colors. And in Organic 1, I had my Organic 1 students actually look at lycopene and adding bromine in water. I'll have that video again available to you. But that's, they changed the color because of the reactions that are happening. So I'm going to say this. I've tried not to be morbid all semester, but I am a little bit on the morbid side. But if you kill someone and you are trying to get rid of the stains, do not simply bleach things. It's not going to hide the fact that there was blood there. You can still find the blood there. It's just not red anymore. So Best thing to do is literally clean it up. Do not just bleach it and get rid of it. That's why forensics people can still find blood if you bleach places and they know that you've done that. So next class, we'll move into the next chapter and do some more problems related to that.